Hello students. Today we will start with immunization topic, which is subtopic of immunohematology. So let's begin. Objectives. We will see active immunization in this lecture. Moving ahead. Now what is immunization? It's the process by which an individual's immune system becomes fortified against an agent. Now what is fortified? Means place provided with defensive works against attack. It means that the body is prepared to defend. The immune system is prepared to defend. Now, as you know, immune system, the adaptive immune system functions when the system is exposed to the molecules, foreign molecules rather, then it will organize an immune response against it. How? Because of the immunological memory. So now if an animal is exposed to an immunogen or the agent, foreign agent, in a controlled way or manner, its body can learn to protect itself. So this is called active immunization. Immunization are of two types: active immunization, passive immunization. So in this lecture, we are going to focus on active immunization. Now, what's the basic difference between immunization and vaccination? So, immunization is the process by which individual's immune system becomes ready to fight against any attack. Okay, and the process is vaccination. And vaccination, it's the act of introducing a vaccine into the body which will produce immunity to specific diseases. Now, most important elements of the immune system that are improved by immunization are T cells, B cells, and the antibodies, B cells. Memory B cells and memory T cells are responsible for swift response, means quick response to a second encounter with a foreign molecule. Just defining passive immunization, it's a direct introduction of these elements into the body instead of production of these elements by the body itself. Which elements I am talking about? These. The elements which are improved like T cells, B cells, antibodies, memory B cells, memory T cells. These are the elements. Immunization is done through various techniques. While defining immunization, I told it's the process by which individual's immune system becomes ready to fight. Process is vaccination. So, immunization is done through various techniques. Most commonly is vaccination. Vaccines against microorganisms that cause diseases can prepare the body's immune system, thus helping to fight or prevent an infection. Let's go ahead with active immunization. Active immunization is the induction of immunity after exposure to an antigen. Antibodies are created by the recipient and may be stored permanently. Okay? Active immunization can occur naturally. How? See, when a person is exposed to a microbe or any other foreign antigen, so his body will produce some antibodies against it. Okay? So naturally, when a microbe or other antigen is received, the person who has not yet come in contact with that microbe, and he must be not having any pre-made antibodies, okay, so he will be producing those antibodies against it. So the immune system of that person will eventually create antibodies for the microbe, but this is a slow process. But if the microbe is deadly, there may not be enough time, or the antibodies cannot be prepared or formed so fast to be used. So next time, the immune response against this microbe can be very efficient. So just remember our childhood infections. We have faced measles, mumps, chicken pox like infections, but only once. After that, our body is immune against them. Now, whole organism vaccine. Whole organism vaccine means live attenuated vaccine or attenuated vaccine. It contains whole bacteria or viruses which have been weakened so that they create a protective immune response but do not cause disease in healthy people. Now, how are they weakened? Now, let's first understand attenuated word. Attenuated means which reduces the virulence, and virulence means the severity or the harmfulness of that pathogen or the virus. Okay, so an attenuated vaccine or a live attenuated vaccine is a vaccine created by reducing the virulence of a pathogen but still keeping it viable or live. Attenuation takes an infection agent and alters it so that it becomes harmless or less virulent. It stimulates a strong and effective immune response that is long lasting. Their function encourages the body to create antibodies and memory immune cells in response to the specific pathogen to which the vaccine protects against the pathogen. Now, common examples of live attenuated vaccines, as I told, childhood infections, measles, mumps, rubella, yellow fever, some influenza vaccines. These are live attenuated vaccine. We have taken it once. Viruses may be attenuated via passage of the virus through a foreign host. Now, what is passage? Passage, the propagation of microorganisms or cells in a series of host organisms or culture media so as to maintain them or modify their virulence. Okay? The initial virus population is applied to a foreign host through natural genetic variability or induced mutation. A small percent of the viral particles should have the capacity to infect the new host. So these strains will continue to evolve within the new host and the virus will gradually lose its efficacy in the original form and lack its selection pressure. Understood? So this is called as passaging or passage. And due to this, virus becomes so well adapted to the foreign host that it is no longer harmful to the vaccinated subject. Attenuated vaccines can be administered in different ways: injection or mucosal, or like orally we take, or in the nasal drops. Injection, subcutaneous. for measles, mumps, rubella vaccine, varicella vaccine, yellow fever vaccine. Second is intradermal. These two are from the skin. Intradermal. Example, tuberculosis vaccine, smallpox vaccine. Mucosal are what? Nasal or oral. Nasal, we take live attenuated influenza vaccine. Drops given directly in the nasal openings. Oral, oral polio vaccine, recombinant live attenuated cholera vaccine, oral typhoid vaccine, oral rotavirus vaccine, directly which is given through mouth. Now let's understand the mechanism. Vaccine function by encouraging the creation of cells, as we discussed. CD8 plus, CD4 plus, T lymphocytes or molecules such as antibodies that are specific to the pathogens. The cells and molecules can either prevent or reduce infection by killing infected cells or by producing interleukins. Live attenuated vaccines also tend to help with production of CD8 plus cytotoxic T lymphocytes and T dependent antibody responses. A vaccine is only effective for as long as the body maintains a population of these cells. Understood? We will not go in detail of the functions, interleukins or the cells involved, but this is the mechanism which vaccines are followed. Now, inactivated vaccine. Inactivated vaccine is also called as killed vaccine. Vaccine consisting of virus particles, bacteria, or other pathogens that have been grown in culture and then they lose disease producing capacity. So, pathogens for inactivated vaccine are grown under controlled conditions and are killed as a means to reduce infectivity, means virulence, and thus prevent infection from the vaccine. The virus is killed using a method such as heat or formaldehyde. In inactivated case, the virus is killed, through two methods, heat or formaldehyde, means using chemical. Inactivated vaccine are further classified depending on the method used to inactivate the virus. Which are those? One is whole virus vaccine. Second is split virus vaccine. Third is subunit vaccine. Whole virus vaccine use the entire virus particle fully destroyed using heat, chemicals or radiation. Second, split virus vaccine produced by using a detergent to disrupt the virus. Third, subunit vaccines are produced by purifying out the antigens that best stimulate the immune system to mount a response to the virus. Okay. Now let's understand the mechanism. The pathogen particles are destroyed and cannot divide, but the pathogen maintains some of their integrity to be recognized by the immune system and evoke an adaptive immune response. When manufactured correctly, the vaccine is not infectious, but improper inactivation can result in infectious particles or the intact particles. Means they will not get divided. Because the killed pathogens in a properly produced vaccine do not reproduce. Booster shots are required periodically to reinforce the immune response. Booster shots are those which are given in a time span, like if 0 months, 6 months, 12 months, after 5 or 4 years. These are booster shots to reinforce the immune response again and again. Injected polio vaccine. Now see, before when I was talking about the oral vaccines, about the polio dose, we give it to the child through the mouth. Drops are given to the child through the mouth. And now here we are talking about the injected polio vaccine, which is a Salk vaccine. Okay, both are different. Hepatitis A vaccine, rabies vaccine, most influenza vaccines, tick-borne encephalitis vaccine. These are the examples. And bacterial, injected typhoid vaccine, cholera vaccine, plague vaccine, pertussis vaccine. These all are given in booster shots. Okay, they are not given once in a lifetime. Thank you. If you have any questions, do ask me.